from Monash University. I study under Dr. Hall at the Hall Lab. And today I'm going to be talking to you a bit about some recent results looking at how male-female differences impact on pathogen fitness. So males and females vary in several ways that can impact on disease. For example, we might have morphological differences between the sexes. And for example, in the blue-headed rats, we know of a parasite that can only infect the ovaries of females, leaving the males immune. Or in the common mouse, we have this behavior of smelling previous markings left by males that causes males to be disproportionately infected with influenza than females. So this is a behavioral difference between the sexes. Or we might know of just inherent immune differences between the sexes. For example, in the red jumblefowl, we know that there is an immunosuppressive effect of testosterone, which causes males to become sicker easier than females. So these are by no means the only three examples, and they're actually quite numerous, but despite that, we actually don't know that much about how male-female differences impact on pathogen fitness. So pathogen fitness can be reasonably broken up into three major components, whether it's the length of the infection, the probability of transferring from one host to another, the top right there, or at the bottom, the proportion of individuals that are actually susceptible to infection. So collectively, these might be referred to as the force of infection. And it's kind of the balancing act, because if one component of the force of infection changes, the others will have an elicited response. But it's not just differences between males and females in their immune-relevant traits, but it is a difference in how they develop or maintain immunity. Specifically, we expect the sexes to invest differently in their immunity, and this might cause a variation in their susceptibility or severity of disease over time. So in this figure here, we have the amount of females that die due to infection with influenza over their age. And we see this pattern of immunity developing with age before being traded off with a reproductive peak, and then the deterioration of immunity with age. And while this is a cool pattern in females, we might expect this to vary in males a bit. For example, we don't always see a reproductive peak in males, and then we don't really expect males and females to age exactly the same. So this would lead us to believe that development of immunity vary between the sexes and also be a factor impacting on pathogen fitness. So what we want to look at then is how do these two factors interact to impact on pathogen fitness? So we might want to look at how host and age interact to affect the length or the virulence of infection. We might want to look at how host, sex, and age impact on pathogen transmission. And as I said, this is really a balancing act, so we want to know how do these two characteristics change relative to one another. To answer this, we're going to use the Daphnia magna Pasteur ramosa system. Daphnia magna is a freshwater crustacean. It reproduces clonally, and males and females are genetically identical, although they do vary in both the lifespan and size, among other characteristics. Pasteur ramosa is a horizontally transmitted bacterial parasite. It mainly causes a decrease in fecundity and lifespan. And we have a really characteristic infection in this system where young Daphnia will pick up spores from filter feeding. The disease will proliferate before expanding in the host. And as this is a horizontal transmission, the host will die, spores come out, the new Daphnia gets infected. So we do know a little bit about male-female differences in the system. Specifically, we do know that among at least two host genotypes, we see that the parasite is able to produce more spores in females than males. So that's really cool. But what we want to do is extend similar knowledge to this to age as well as sex. So what we're going to do then is, or what we have done, I should say, is we infected both male and female Daphnia with either a parasite that produces a small amount of spores, referred to as low growth, or a parasite that produces a high amount of spores, high growth. They are the same parasite, Pasteur ramosa, just two different genotypes with different disease dynamics. And then we're going to expose them at one of 4 age groups, trying to capture aging in this system. And then we're going to measure traits that are similar between males and females, or I should say are able to be measured similarly between males and females, like how many spores are produced in each host, which would be our measure of transmission. And we're also going to measure how long the infection lasts in males and females, our measure of virulence. So to get at this first question, what we did is we put together some mortality curves to look at how the rate of mortality varies due to sex and age. And what we find is a significant interaction between age and sex, And what we see is in females, the rate of mortality increases with age. And then we have a really big difference in terms of the proportion surviving between those infected at a young age, day 10, and those infected at old age, day 40. Conversely, in males, what we see is the rate of mortality doesn't really change regardless of when you've been infected. And we might be able to get at this a little better by looking at how how we see a change in lifespan between males and females due to infection. So again, we see a significant age by sex interaction, and what we have plotted here is a change in lifespan. Positive indicates a decrease in the amount of days as compared to the controls. And what we see is, in females, the parasite is always virulent. We're always having we're also having a decrease in lifespan due to infection. Whereas in males, the parasite's only virulent at one age class. So this helps us get at a little bit of why we see an increase in mortality in females versus relatively flat rate of mortality in males, and maybe suggest that aging due to infection varies between the sexes a little bit. So if age and sex impact on the length of infection, now we want to look at how do age and sex impact on the pathogen's ability to transmit. As I said earlier, we measured this by the amount of spores, which is something we measure at host death. We just simply count the amount of spores in the host. And we have a parasite by sex interaction. And what's really cool is in males, we actually don't see the amount of spores really change much over time or parasite genotype. At the very end of the infection, they were infected with no mature spores, but beyond that, it's pretty similar. And this is in stark contrast to females, where we actually see the two different parasites give us really different results. We see, as expected, the high-producing parasite was producing more spores than the low-producing parasite. This actually trades off with age, and we see that as the host ages, we see less produced in the high-producing parasite and the same along age with low-producing. So I guess like a quick recap is, we see differences between two parasite genotypes in females, but we don't see these differences in males. So if both age and sex can impact on two really big components of parasite fitness, what we next want to look at is how do these two interact with one another? Force of infection is a balancing act, so we might expect these to vary, as one of these to vary as the other does. So to do this, we plotted the amount of spores produced against the change in lifespan. And just to emphasize a couple of trends that I think are interesting, I just drew a couple of lines over this for you. So what we see in males is that regardless of the age that it was infected, we don't see any variation in spore length. We're always going to get a similar amount of spores produced no matter what age they are. And this means that a parasite infecting a host at day 10 is much more fit than a parasite producing spores in a host at day 30. Because they're producing the same amount of spores, but killing the host and moving on to the next generation quicker. In females, though, what we see are really strong differences in the pattern, or where the most variation in infection is. In a parasite that produces a high amount of spores, we see only real amount of, so these are the triangles here, we only see variation in the spore load, the amount of spores produced due to infection. Whereas in the parasite that produces the low amount of spores, the circles, we really only see variation in the change in lifespan. And this is another example of us only seeing differences between parasites within females rather than males. And this means that the force of infection is fundamentally different depending on the host that you infect. We will only see really big differences in females than in males in this system. So altogether then, we've shown that host sex or age can impact on two major components of parasite fitness, as well as the interaction between them. And the reason this is important is because host sex and age are basic characteristics of natural populations. We will see between two populations, not only differences in the amount of individuals in each age class, but also the proportion of males to females in each age class. And that means that if the things that we've seen in the system hold up in others, that variation between populations and variation over time are going to be two major characteristics to determine before we understand how disease can spread or impact a real population. So with that, I would like to thank tons of people who have helped me out, including Monash University that paid for my trip over here, and Savannah Ganeshow for illustrating my talk. Thank you. I think I left a lot.
So what about social interactions? How does that influence transmission between, say, males and other males, or males and females that might connect these patterns?
I don't want to say we, but I know personally I don't know much about social interactions with apnea, but I would have to say that in a natural population that would be really important to consider, because males aren't a very common sex of apnea. I induce these in the lab. So in reality, we will probably be seeing transmission between females more often than between males and females, for example. It would impact how we would expect them to spread in the system, for sure.
So, if I understand right, it looks like infection increases lifespan in both males.
Yeah, so
what's going on with that?
It's given me a lot of thought, but my gut reaction is it's a product of the sampling design. So we just set up a large pool of females and males, and as we needed to infect some at day 30 or day 48, for example, we pulled 60 or 70 males. So I think we are artificially potentially picking the hardier males at the end of the experiment. It's unfortunately a necessary part of the design because males and females have such a different lifespan that if we were to pre-allocate males, especially at day 40, we simply wouldn't be infecting very many at the end of the experiment.
This is a probably dumb question. So you have males and females that are like both clonal, right?
Yes.
So how does sex determination work, and what's the effect of being male on the same clone?
So sex is an environmentally determined characteristic in this species. It comes about due to a variety of stressors in the environment, like salinity or differences in daylight cycle. And sorry, the second part was what does it mean to be a male in the system?
No, what's the, genotypically, like how, if they're genetically identical, then how does the male phenotype differ from female?
Phenotypically, so they do have a different size and morphology. They have this kind of tweezer appendage on their face to attach to a female. I imagine that all is still simply kind of an expressed response to its environment. All right, well, I might go sit down. Thank you very much.